Hello and welcome to my channel. And as you all know, this is all about community medicine and making it more approachable, comprehensible by all students and teachers alike. Well, this is a continuation of my previous presentation, basic statistics, and taking it to a different aspect of statistics, that is the measures of central tendencies, that is the summary statistics. One side we'll be talking about the central tendencies, another we'll be talking about the measures of dispersion. And along with it, we'll also try to understand the concepts of central limit theorem along with the normal distribution. So let's go ahead with it. But if you recall, biostatistics is the application of statistical methods to medical, biological, and public health related problems. It is the scientific treatment given to the medical data derived from a group of individuals or patients. To remember easily, find this mnemonic to your use, that's CPED. So, in fact, you can even remember it as CashPED, CPED. C stands for collection of data, P stands for presentation of the collected data, A stands for analysis of the data, and I stands for interpretation of the results, and D is the decision made on the basis of such analysis. And these are the basic components that biostatistics looks into. And today we will be talking about the summary statistics. It is a part of descriptive statistics that summarizes and provides the gist of information about the sample data. The statisticians commonly try to describe and characterize the observations by finding a measure of location. That is, how it is located in terms of its position. It can be how it is more located towards the central area through the central tendencies, or through the rest of the area through measures of non-central tendencies, and how spread out are they from the central value, that is in terms of measures of dispersion. Now coming to central tendency, the basic measures of central tendency are mean, median and mode. In the mean you have arithmetic mean, geometric mean and harmonic mean, but mostly when we talk about this in context of our examination and understanding, we generally refer to arithmetic mean. So what is this mean? This arithmetic mean is nothing but the sum of all observations in a data divided by the count of all observations in that series mathematically. It's also called the average and is the most popular and well-known measure of central tendency. It can be used both discrete and continuous data. Although its use is most often with continuous data. So if we have a n number of values in a data set and they have the values like x1, x2 and so on and so forth to xn, then the sample mean will be usually denoted by x bar. And this x bar is nothing but x1 plus x2 plus such and such to xn divided by n, where n is the number of observations. So sum of all the observations divided by the number of observations, and it is usually expressed in the form of summation x by n. Medians. In a series of observations, the median is defined as the middlemost value. When it's a discrete series, if the count of the observation is odd, the median is simply the n plus 1 by 2th value. That is that observation in the ordered series. However, if the discrete series has an even number of observations, then the median is approximated as the arithmetic mean of the n by 2th and the n by 2 plus 1th observation of the ordered series. For example, if there is a series of observation 8, 12, 151, 7, 5, 10, 8, 9 and 11, if you order it, it becomes 5, 7, 8, 8, 9, 10, 11, 12 and 151. This series has 9 observations, hence 9 plus 1 by 2, 5th observation is the median. And here the 5th observation is 9. Now say we had another observation, say 6. In the new ordered series, then it is 5, 6, 7, 8, 8, 9, 10, 11, 12 and 151. The series now has 10 observation which is even. So the median is the arithmetic mean of the 10 by 2 equal to 5th and 10 by 2 plus 1, that is 6th. So basically the median emerges to be 8 plus 9 by 2, that is 8.5. Now coming to mode. The mode is defined as the value which is most frequent in a series of observation. And how do you do it? In a simple series, supposing 8, 1, 10, 8, 3, 7, 4, 8, 9, 6. Which one do you find to occur most frequently? It is 8 here. It's occurring 3 times. There can be multiple modes or even no modes in a series. For example, 9, 11, 10, 11, 9, 13, 14, 12 has 2 modes because we have 2 times 9 and 2 times 11. So whereas a series like 2, 9, 5, 2, 9, 5 has no mode at all, as all the elements have exactly the same frequency. A mode can be calculated by observing a data series. So let us look at the advantages and disadvantages of this central tendencies. Well, coming to mean, the advantage being all observations are considered for the calculation and further algebraic treatment is possible. But the disadvantage being it is particularly susceptible to the influence of the outliers. These are the values that are of unusual value, like in the previous slide we mentioned, 8, 9, 10, 11 and then 151. Here 151 is the outlier because it's an unusual value compared to the rest of the values. So either that value is too small or too large in numerical value. And mean is very susceptible to be affected by this outliers. Now coming to median, it is easy to calculate and it is not affected by the outliers. But the disadvantage being it is not based on all the observation and further algebraic treatment is not possible and is highly affected by the sampling changes. Coming to mode, the advantage is it is also not affected by the presence of outliers. But the disadvantage being further algebraic treatments are not possible and for some series mode may not be defined also. Now let us move on from the central tendencies to the measures of dispersion. The dispersion is nothing but looking at the scatter of the data from the central position. And for these we use different measures of dispersion like range, variance, standard deviation, semi-interquartile range, coefficient of variation and standard error. So the dispersion is basically a statistical measure that indicates how the observations are spread out or scattered on each side of the center. If the value of the dispersion is small, it indicates high uniformity of the observations. And if the absence of dispersion in the data indicates the perfect uniformity. So this situation arises when all the observations are identical. And what is the importance of these measures of dispersion? The knowledge of dispersion is vital in understanding of statistics. It helps to understand concepts like the diversification of the data, how the data is spread, how it is maintained, and maintaining the data with a central value or central tendency. Moreover, dispersion in statistics provides us with a way to get better insights into data distribution. So as you can see, the variance, range, standard deviation, coefficients of variance are all measures of dispersions. Range is the measure of the difference between the largest and smallest value of the data variability. Range is the simplest form of measure dispersion in fact. Supposing we have a data series 1, 2, 3, 4, 5, 6, 7. The range will be the highest value minus the lowest value. That is, here the highest value is 7 and the lowest value is 1. So the range is 6. Now coming to interquartile range. The quartile segments may be distributions that order from low to high into 4 equal parts. The interquartile range or IQR contains the second and third quartiles or the middle half of your data set. As you can see in this picture, between the Q1 and the Q3. Whereas the range gives you the spread of the whole data set, the interquartile range gives you the range of the middle half of the data set. Now, another measure of dispersion is the variance. In simple terms, the variance can be calculated by obtaining the sum of the squared distance of each term in the distribution from the mean and then dividing this by the total number of the terms in the distribution. It basically shows how far a number, for example, students mark in an exam is from the mean of the entire class. The formula is sigma square is equal to summation of x minus mu whole square divided by n. Where mu is the population mean, n is the total number of observation, x is the observation in concern. Now, coefficient of variance is a measure of the relative dispersion. It is used to compare variation of distribution with different units relative to their means. So, when we have differing units, that the units are not similar, that is where we use the coefficient of variation. And this coefficient of variation is equal to the standard deviation divided by mean multiplied by 100 and is usually expressed in terms of percentage. Now, let's understand standard deviation, one of the major tools of measuring dispersion. See, if you look at these three clouds, in one, all the numbers are identical, 7, 7, 7, 7. In the second cloud, there's a little more variation. In the third cloud, the variation is more pronounced. So, in the first cloud, there is no deviation. In the second cloud, there is a minor deviation. But in the third cloud, we have an obvious deviation. Well, the standard deviation is actually a statistic that measures the dispersion of a data set related to its mean and is calculated as a square root of the variance. The standard deviation is calculated as a square root of the variance by determining each data point's deviation relative to its mean. So how do you do it? What's the process? You calculate the mean of your data set, then subtract the mean from each of the data values and list the differences. Square each of the differences from the previous step and add the squares from the previous step together. Subtract 1 from the number of data values you started with. Divide the sum from step 4 by number from step 5. Take the square root of the number from the previous step. This is the standard deviation. For this, it is very important for you to understand the central limit theorem as well. It is the basis on which that distribution that we are talking of is measured in statistics. In research, we get a good idea of the population mean. Ideally, you would collect data from multiple random samples within the population. And a sampling distribution of the mean is actually the distribution of the means of these different samples. The central limit theorem tells us that the law of large numbers, as you increase the sample size, that is the number of samples, the sample mean will approach the population mean, because then it will get closer to the real value. And with multiple large samples, the sampling distribution of the mean is normally distributed, even if your original variable is not normally distributed. So as we are talking of this normal distribution, let us try to assess what is normal distribution. Well, the key characteristics of this distribution, that is how spread are these observations, there are certain key characteristics that we spot in the graphs. That is, the mean, median and mode are exactly the same in this kind of distributions. And this distribution is symmetric about the mean. Half the values fall below the mean and half above the mean. The distribution can be described by two values, the mean and the standard deviation. So in a normal distribution, the data is symmetrically distributed. There's no skewing, as you can see in the graph. And when plotted in a graph, the data follows a bell shape, with most values clustering around the central region and tapering off as they go farther away from the center. The normal distributions are also called Gaussian distributions or bell curved because of their shapes. Now, what is a standard normal distribution and how is it different from the usual normal distribution? In the standard normal distribution, which is also called the Z distribution, it is a spatial normal distribution where the mean is 0 and the standard deviation is 1. Every normal distribution is a version of the standard normal distribution that has been stretched or squeezed and moved horizontally right or left. So if that is so, what happens here? While individual observation from normal distributions are referred to as X, Here they are referred to as Z in the Z distribution. Every normal distribution can be converted into the standard normal distribution by turning the individual values into the Z scores. So instead of the sample means, we are plotting the respective Z scores. And the Z scores tells you how many standard deviation away from the mean each value lies. So here you see a standard normal distribution. And how do you calculate the Z? It is calculated by measuring the deviation from the mean from the population mean divided by the standard deviation. And each Z score is associated with a probability of p-value that tells you the likelihood of values below the Z score occurring. So if you convert an individual value to a Z score, you can then find the probability of all values up to that value occurring in a normal distribution. And one of the important aspects that you should be aware of about the normal distribution curve and understanding the concept of standard deviation in context of it is how, seeing a normal distribution curve, you can comment on the standard deviation of that particular data series and what percentage of the real values fall within that. Well, mu plus minus, that is the mu is the population mean, plus minus 1 standard deviation, we have the 68% of the real data falling within it. When mu plus minus 2 standard deviation, we have 95% of the data. Mu plus minus 3 standard deviation, we have 99.7% of the data falling within it. Now, what is standard error of the mean of standard error? It is a measure of variability among the means of samples selected from the certain population. Standard error of the mean is S by root over n. Here S is the standard deviation. So how is standard deviation and standard error different or similar? Well, standard deviation implies a measure of dispersion of the set of values from their mean, and standard error connotes the measure of statistical exactness of an estimate. Here, standard deviation is majorly descriptive statistic, and standard error is largely inferential statistics, and it measures how much observations vary from each other, while standard error precisely sees the sample mean to the true population mean. Distribution of the observation concerning normal curve is seen in standard deviation, and distribution of an estimate concerning normal curve is seen in standard error. The formula being in standard deviation the square root of variance, and the standard error being standard deviation divided by the square root of the sample size. If we increase the sample size, it gives a more specific measure of the standard deviation, but it decreases the standard error. So, a quick recap regarding the measures of different tools of statistics. The range is belonging to the descriptive statistics, and it looks at the spread of the extremes of the data, and the formula being highest data point minus the lowest. The standard deviation again is a part of the descriptive statistic, and typically, or roughly speaking, average difference between the data points and their mean. That is, SD is equal to root over of summation of x minus mu square divided by n minus 1. The standard error is inferential statistics part, and it's a measure of how variable the mean will be if you repeat the whole study many times. So standard error is standard deviation divided by root over n, where n is the sample size. And confidence interval, that is usually taken as 95% in the different research methodologies, is also part of inferential statistics. A range of values, you can be 95% confident it contains the true mean values. Well, that brings us to the end of this second part of the basic statistics, where I briefed you about the different central tendencies, the measures of dispersion, the basic concepts of normal distribution, and how would you relate to this normal distribution curve and interpret the standard deviations and make a conclusion based on that. Hope this is of help, and remember, these are all important from your point of view of your theory, your practical, your viva. So happy studying, and if you do like the video, please like, share and subscribe, and press the bell icon to stay tuned for more academic contents. Thanks once again.